All right, well, yeah, so thanks for having me. I thought I would talk about kind of a mixture of the work I've been doing that hopefully kind of sits at the nexus of what this seminar is about, which is kind of in between planetary science and the geophysics side of things, and more of the astrobiology side of things. So I thought I'd take a chance to kind of talk about how life gets entrained into the ice, and once it's there, um, what's really going to control how easily it can be there. And I think this is an important topic because pretty much everywhere that we're looking for life across the solar system is going to have ice in it. So whether that's the icy moons like Enceladus or Europa, excuse me, or Titan, or even the Mars system. And I think one of the big advantages is that we have a lot of different environments on Earth to kind of use as proxies and analogs for these systems. So kind of probably the biggest and widest known one is sea ice. And so in this system, the idea is basically you're going to have this layer of ice, and then at the base of this ice, instead of having kind of this abrupt boundary, you're going to have this kind of porous system with all these pockets and channels where small creatures and things are going to live in there. And this is encouraging because beyond this cartoon, we can go and we can take ice cores and we can look and we can actually see that in this lower layer, you have this kind of abundance of life right at the base. And when we zoom in even more, this is an image, it's like a micro CT or a CT scan of sea ice where the white areas are these small brine channels and things like that, and the negative space will actually be the ice. And if we keep zooming in, you can kind of get an even better idea where the reason this is happening is because you have these ice crystals, and in between there, you're going to have this supersaline brine where kind of all of that residual salt and all of these residual organisms are going to get rejected and kind of live and thrive. If we keep zooming in, you can actually see, these small algae in these brine pockets and channels in actual natural sea ice. So this is kind of the systems that we're talking about. Some other systems are underneath ice shelves, and so these are some images of some larger macrofauna underneath ice shelves. On the left side is beneath the Ross Ice Shelf, which is kind of near the front of the ice shelf, so this is going to have more interactions with the open ocean. And then on the right side, kind of a more local, is Brittany and crew a few years ago were down in Antarctica and saw similar things underneath Thwaites near the grounding zone. And so in this system, you basically kind of have this clear ice base at the base, and then there's kind of this secondary shelf where if one of those guys, if I'm butchering this, want to jump in and correct me, but you kind of have these two-tiered systems, and so these anemones are kind of living in this lower ice layer and thriving there. And this is way far back from the front of the ice shelf. So kind of understanding how these things can thrive in these environments. Another system that I look at a lot is lake ice from hypersaline lakes up in British Columbia, Canada. And so on the left side here is basically just a chunk of ice that we extracted from there. And so the bottom of the ice that's going to be in contact with the lake water is on the left side, and the top of the ice is going to be on the right side. And so you can see just by the color that you're going to have different organisms and algae in these systems. And on the right side is going to be fluorescence microscopy of kind of these layers as they go down. So in the top, you're going to have less biology, and as you kind of move down towards the base of the ice shell near the bottom, you're going to have more and more biology kind of living in this system. And just kind of round these out, I want to talk about not exactly what I'm going to be talking about today, so I'm going to be focusing more on kind of this oceany environment, but also fern and snow and permafrost are super important and have big impacts with biology, and also cryoconite holes, which if people haven't heard about these, are these really cool systems where you basically get soil and dirt that gets blown in on glaciers, and they eventually will melt and form these little melt pockets that kind of have their own little ecosystems in each pocket. And then they'll freeze during the winter and then remelt in the summer, and it's kind of these really interesting cycles, and people have done some really cool microbiology work on those systems. So then to move to kind of like the planetary system, so going from what we know about life on Earth in these systems to potential systems in the solar system that might be good for life are things like ice ocean worlds. So on the left you have a picture of Europa, where you basically are going to have this thick, maybe 100-kilometer ocean, and then over the top of that you're going to have this super thick ice shell, maybe 10 to 30 kilometers. But in this ice shell, it's probably going to be super dynamic, and you might have all these pockets and fractures and all these kind of like different processes where you could potentially have liquid water at different points within the ice shell. And that's really what we're looking for, this whole kind of follow the water idea. We're looking for these areas of liquid water. The nice thing about having them in the ice shell is they could potentially be more accessible, but there's also a lot of processes in there that could kind of generate chemistry in environments that might be better than even just the open ocean, which is kind of akin to what we see in sea ice on Earth, because that layer at the bottom of the ice actually has a lot more biomass than anywhere else in the water column in the Arctic oceans. And then for Martian environments, the top picture is going to be something that's kind of been suggested of having like paleo lakes. So this would be more biological systems in the past. But then at the bottom, then there's also situations now where people are saying that there's these potential lakes beneath the ice caps and things like that. So that's a system that could exist now, but could have also existed in the past if Mars was glaciated at different times. So these are all kind of different ice ocean, ice water systems that we're thinking about when we're talking about life in the ice. And so kind of what are the good things, what are the bad things about this? Why does life like to live in the ice? And so one of them is just water availability, right? So if there's ice, there's going to be a potential to have liquid water, which is something that is going to be associated with life, so it's a good place to be. The second is that it's porous substrate, so instead of just kind of freely floating around in the ocean or in water bodies, the ice kind of provides this location for organisms to be protected, but also to latch onto and kind of thrive in. So I kind of think about it as this kind of inverted benthic system. So, the sediments of oceans have a ton of biology in them. So it's basically what's happening there, kind of in an upside down situation. Like I said, sea ice has this huge biomass in it. And so in these planetary environments, these are kind of locations that we also think could have a lot of biomass in it. And one of the reasons for this is that when you have this kind of concentrated system, when all of these salts and potential nutrients are kind of getting pushed into these veins and channels, it kind of is going to, make this super rich brine left over the biology can take advantage of for things like metabolism. And I think this cartoon at the bottom does a good example of kind of showing that, everything kind of got pushed out away from the ice and into this kind of residual pocket. The cons to this, though, is also surprisingly very similar to the pros of this side. So one of the things is that water availability. So even though there's brine in these pockets and channels, in a lot of these systems, something like sea ice or the ice shell, there's still going to be this progressive freezing front. So eventually it will get cold enough that even this brine is going to solidify out. So if organisms don't have some way to either be mobile and kind of move to a better situation, or some kind of chemical defense against that, a lot of these organisms excrete this EPS, which is kind of like this antifreeze macromolecule that they produce to kind of keep things brine right around where they are. Or if they can actually get trapped in and frozen into the ice and kind of just put into stasis and then, become viable once conditions become more favorable. Another side of the cons is that when these brines get concentrated, there's benefits because it does kind of concentrate potential nutrients and chemical gradients and things like that, but that can also become an issue. If these brines get too salty, their ionic composition just gets too high that, it's not beneficial for the organisms anymore. It can also change the pH and the water activity of these brines as these things kind of crystallize out and get more and more concentrated. So that can start to kind of become detrimental to organisms and kind of decrease the habitability of the systems. And so this is kind of a look at some of the work that I've done, that we've done up in Canada, trying to look at this. And so this profile on the left is basically looking at ionic profiles in ice cores that we've taken, as well as basically the cell counts or the biomass concentration in that ice cover. And so what you can see is that in that shallow ice cover, the bulk quantities of those ions are going to be lower, but you have to remember that it's also going to be colder at the top. So what's ever going to basically be residual brine and still in solution will actually be probably a lot more concentrated than what's at the bottom, and that's going to be reflected in kind of the amount of cells that are going to be there, right? Like you can see that there's a definite trend towards the bottom of those ice cores where the biomass is going to increase with depth. And another way to look at that is these right plots are basically taking the brine chemistry from these environments and then freezing them out through temperature. And so you can see in the bottom right plot, you're basically looking at temperature versus the solution volume. So once you hit its freezing point, it's basically going to drop down. You're not going to have much fluid left. And when that happens, on these left two plots, the pH is going to move way far away from neutral, and that water activity is going to start to drop. So these are kind of those issues that's probably going to happen in these brines. Excuse me, and that's going to potentially make these systems less favorable for any of the organisms living in there and kind of decrease the habitability of these systems. So kind of the big questions here that kind of guide a lot of my work is in these ice brine systems, what's going to determine kind of how they evolve over time, both physically and chemically? And the second question is how does that chemistry and the fluid transport and things like water activity and pH actually limit the habitability of these systems? And finally, how does that and what we know about those environments, the habitability of them and how they evolve through time on Earth, kind of guide us in looking for the best places to look for habitable environments on other worlds? And so the way that I kind of go about that is two ways, which Brittany kind of mentioned, was kind of modeling work, but also looking at these terrestrial analogs. So kind of to start from the beginning is some of the things that I've talked about is not looking at ice as this kind of solid block of ice that we think about when, you know, we put ice in our freezer to freeze out, because that's more the freshwater side of things. And in that case, you're going to have this kind of like pure solid block of ice with not a whole lot going on. Excuse me. On the right side is what we think about in the saltwater system, which is probably going to be more relevant to just about any planetary system. And in that case, you kind of have this two-phase system where you can see in this block of ice, you know, this food coloring that got poured on top of here is kind of permeating into these pockets and channels, and you can have all this fluid and solute transport happening throughout this ice as long as it's basically above the temperature, its eutectic temperature, where everything is going to freeze out. Another really great way to think about this is to kind of use the analogy of a magmatic system, and I think it's a really good thing to think about because now people are starting to talk about like cryovolcanism on icy worlds and stuff like that. And I think that's a really good way to think about it because it's kind of a similar system where you start out with one composition, and as you cool it down, you're going to hit these points where different things are going to kind of start to solidify and crash out of the system. And in the ice system, that's basically just going to happen upside down, but it's going to be over a smaller range. And so what happens is you basically, as you're forming this ice, in something like sea ice or a planetary ice shell, you're going to kind of form this mushy, slushy layer that has like an ever-evolving composition as you're freezing out more and more ice as that layer thickens. And the picture on the right is actually an image of laboratory-grown sea ice where you can kind of see this actually occurring, and you have the pure fluid sitting down at the base, and then you have all these channels and pockets and kind of this two-phase system where it's very obvious that there's still fluid and still solutes and still components that are going to be in this ice, and it's really going to determine, you know, what that ice looks like and how it evolves. And so a lot of my work in the modeling side of things has been focused on trying to model these systems. And so the most recent stuff I've been doing is trying to move to kind of like a full 2D view of what this will actually look like and can we model that system. So I've been using this model called Softball written by Jamie Parkinson. And so kind of the idea here is we're going to start with just this block of fluid. That's kind of our little square world that we live in, whether it's an ocean or a lake or something like that. And so at the surface, you can kind of set a specific temperature or a temperature gradient or something like that. The sides are periodic, so it's kind of nice. Anything that flows out of the right side comes in at the left and vice versa. And then basically the governing equations here are going to be conservation of mass at the top. So that's basically just saying we can't create or destroy any fluid. Typical Navier-Stokes kind of stuff. The second one is going to be Darcy's law, which basically describes how fluid's going to flow in porous media. So as we freeze out this ice and it becomes this kind of porous thing with all these pockets, this is going to be basically your fluid velocity equation that's going to govern that system, which is going to be conservation of momentum. That next line is going to be conservation of energy, which basically says how temperature is going to change in our system is going to be governed by the first term, which is basically advection of temperature throughout the system, advection of heat, sorry. The next one's basically just diffusion of heat, and the last term is going to be phase change. So when ice forms, it's going to produce heat, and when it melts, it's going to take heat. The last one is conservation of salt, basically. So how the salinity of the system is going to change is going to depend on advection as well, which is the first term, diffusion, which is the second term, and then a phase change term as well. So when you form ice, your brine is going to get saltier, and when you dissolve ice, it's going to get less salty. And so this is kind of the end result of that. So what we're doing here is kind of simulating that system where we start out with this fluid box and we cool it down from the top. And what you can see is basically as that ice front propagates down, this is looking at the concentration of the system. You can see these pockets and channels form, and you can see kind of the downwelling of a lot of the salt from the system into the underlying ocean. And so there's a lot of blue in this picture. And so the blue, if it's less than white, is basically going to say that the blue regions are going to be less salty, and then the red regions are going to be more salty than your original ocean composition. So overall, if you average this, it's going to be less salty than the underlying fluid because you're rejecting a lot of properties. But it's these small regions, these small pockets that have these high concentrations that are going to be of interest and are probably going to be where things are living. So to take another view, this is the same simulation, but instead of looking at concentration this time, we're looking at porosity. So this is basically how much liquid is left in the system, where you can see yellow is this kind of big ocean underneath. And the top, this dark blue region, is basically where you've hit that eutectic point. You've hit a temperature where everything is going to be frozen out of the system and kind of locked in there. And they're kind of look at a snapshot of these environments as that's happening, zooming in kind of at this boundary layer. So the rows in this are going to basically be at different depths in that simulation as things were getting frozen out. The left column is going to be the liquid fraction, what we were just talking about. The center component is going to be the brine salinity, so basically how saline the brine is in these different regions. And then the right column is going to be the salinity gradient, which is basically like the change in salinity. So that's one of the things that we're really interested for from like a metabolic side of things, to say, you know, these large salinity gradients in these regions could be something that could be taken advantage of by different microbes. And so when we take a closer view at one look of these, one of the things that people have suggested is at these interfaces, at these regions where you're having these large salinity gradients, is you could have a system that would almost be like an upside down chemical garden that we would see on Earth, where you have this huge drastic change in salinity across an interface, which is something that can be taken advantage of for life. The bottom left is these brinicle systems that are produced down in Antarctica that were kind of suggested by Steve Vance, that this would be something that even looks kind of like these systems. And we can actually produce those in our models. You can see in that top left image that we produce those. One of the things is they really only happen in low current environments, so that might not be a good situation for ice ocean worlds, as they're believed to have a lot higher currents. But one of the important things to note about this is on that top right image, the salinity gradient is actually going to be the highest in the ice. So you can see there's kind of this sharp-ish interface. Not to contradict myself, but in that brine channel is where that actual highest salinity gradient is going to be. So it might actually be internal regions in the ice and not these actual structures that are moving down that would be the best places for organisms in these systems, these super high salinity regions. And this is just another snapshot of kind of the same thing, which is just, you know, kind of a sanity check to say, does this match the observations? Are we doing a good job, right? And so the left is the model of that liquid fraction, and then to the right again is that laboratory-grown sea ice situation. And these channel structures and this gradient porosity is really, really well produced by the model. So we're really happy that it's basically simulating these thermochemical dynamics as these systems are evolving, so we can make good predictions about these systems. So one of the things that I've been looking at a lot lately is kind of like, okay, this works well and we can match it up to what's going on Earth, but what does this mean for icy worlds? And so one of the things that we found is that in ice ocean world systems where you have these thicker ice shells, is that interface is going to basically grow with the ice shell. So these panels basically just increase in size from about 10 meters per box to 100 meters on the right side, and you can see that interface is basically growing as the ice thickens. And so this is really important because we can kind of come up with this simple relationship that shows that it's going to follow that point in the ice shell where everything is going to freeze out, this eutectic point. So this is really interesting because we found that it's about 5% of the ice shell thickness. So if you have something like a 30-kilometer thick ice shell on a moon like Europa, that means that this kind of porous region where you could have this fluid flow and fluid present could be up to like 1.5 kilometers thick. And so this is really interesting because instead of having this few centimeter thick system that we see at the top right in something like sea ice, you could have something like that that could be much, much thicker. So you would have just a lot more real estate, a lot more space, a lot more variety and heterogeneity for things to kind of populate and work their way into and thrive in. And so I think that does a really good job. The model's done a really good job of describing how these systems are going to evolve. And so kind of the next question number 2, is how do we relate what we're seeing in these models, so something like these salinity profiles or these porosity profiles, to actual habitability, like how habitable are these systems, which is kind of the next hurdle that we're hoping to do with the model, because right now salinity is kind of non-specific. It's kind of talking about concentration, so we give it some initial concentration of salt, but we don't really specify what salts they are, which can be really important for determining basically how well organisms can exist in these systems. So things like water activity and pH are going to definitely be determined by what ions are there, what salts are there. And so that's kind of the next thing, is saying, how do we nail the specific geochemistry so we can not only say, well, these are very concentrated environments, but this is going to exactly be the composition in the chemistry, and this is how potent or non-potent it would be for organisms in this system. And so this is some of the field work that we've been doing to try and hone in on that. And so one of the big ideas is that we have a pretty good idea of how sodium chloride systems work from sea ice, but the reality is that a lot of different oceans all over the solar system might be very different compositionally than just our sodium chloride ocean. So we've kind of honed in on these lake systems up in British Columbia because they offer kind of this huge spectrum of chemistries that we can go test and look at ices formed from all of these different systems to kind of learn how those specific ices evolve over time. And so these lakes are kind of dotted all over central British Columbia. What's really nice is there's a huge spectrum of salinities. They can be almost fresh lakes or saturated. Some of these lakes will actually just have layers of salt hydrates on the bottom of them, and I'll show some pictures of that in a bit. What's also nice is there's a lot of different chemistries. There's magnesium sulfate lakes, sodium sulfate lakes, and some sodium chloride and calcium carbonate lakes as well. So you can kind of go up there and pick your favorite analog lake in whatever salinity range you want to kind of look at how that system evolves biologically and physically and things like that. So kind of the goal of this project is to go up and do just that, is to look at a bunch of these different lakes. And we've gone up in the summer and the winter to kind of look at how they evolve seasonally as well, but to kind of get a full spectrum of how these ice systems evolve and how that depends on their chemistry and things like that, and also get full profiles of what the ice looks like. So honing in on things like composition and biomass, pH, stuff like that. How does the structure of this ice evolve? With the goal to basically build up this catalog that says, this is how the ice of any system you can think of, not any system, but a lot of different systems, so you can compare that to models and also try and forecast that into, different planetary environments. And so these are some of the results from this last season that we went up in February of 2020, which feels like a long time ago now. And so these top ones are some of the ones that I'd shown before, these magnesium sulfate systems, where I'm showing both the ionic profiles that kind of have this C-shaped curve, and also this kind of gradual increase in cell density as you move down through the ice column. This bottom left panel is also a magnesium sulfate lake system. But what was really interesting about this site that we sampled is it had an internal brine layer. So there was probably 5 or 6 centimeters of ice, and then there was actually like this pocket of brine that we like used a syringe to basically pull the brine out, and it was kind of a finite region. And below that there was more ice, and then at the base you got back to like the actual lake brine. And so you can see this huge spike in this region in the center of this ice core, basically in the cells and the ion chemistry where this was melted out. That basically just shows that, you know, when you have that fluid availability, it's going to be populated by cells, even if it's in the middle of an ice core or something like that. And then the bottom right one is going to be a sodium sulfate lake that we went to as well. Unfortunately, which I'll talk about in a bit, that lake was a little difficult. It was a little warm that day, so the ice was basically super, super porous and was very, very hard to sample. Some of the other things that we did up there is we put up these temperature sensors to basically get seasonal profiles of the temperature of both the air and actually in the lakes to basically know how to force numerical models. If we want to try and simulate these, we need to know the air temperature that's forcing these systems, but also to look at how the brine temperature kind of evolves throughout the system. So at this lake, when we were there in the summer and put in these temperature sensors, there wasn't any salt on the bottom of the lake, and then throughout the winter, as the ice started to form and saturated the brine underneath, because it's going to convect all that salt down underneath, it actually started to precipitate salts out at the base of at the base of the lake. I mean, you can actually see this, we think, in these temperature plots of the red and the yellow. These were in the brine, one above these precipitated salts and one below them when we went and pulled them out, but there's a lot of variation, and then you can see them kind of bottom out and then pop back up and just kind of remain stable at the same temperature. So what we think happened there is basically this brine super saturated, and then once these precipitates start to form, they release latent heat and then basically keep the brine at a constant temperature. And this is just kind of an image of those salts that we pulled out of the bottom of the lake. So we thought it might have just been ice that was oddly forming at the base at first, because it kind of has that same crystalline look, but these are actually two types of different salt layers that occur at the bottom of the lake as they kind of precipitate out of solution throughout the winter. So what we can do is basically take these profiles, something like the ion profiles. If we add them all up, we can get back that bulk salinity that is modeled by our models currently, and kind of match up things like these bulk salinity profiles with the model to see if they're doing a good job. And in a lot of cases, they do pretty well. So this is forced with that temperature data that we have, and we can basically simulate how those systems are evolving. So we're happy that it's, you know, kicking back out that chemistry from the bulk sense, even if it's not necessarily being able to do all the species just yet. And then this was also that temperature jump that we were looking at. So we were able to basically add in a precipitate into the model and show that if we don't use that precipitate in the model and we don't consider that salt hydrate precipitation, we cannot actually reproduce the temperature profiles. But if we do include that precipitation, then we can, which is really interesting, because if there wasn't that precipitation event, it basically just suggests that the temperature of the brine should have just kept dropping and dropping. But it basically can prolong the lifetime of these brines just because it is this source of latent heat, which you can see come in and kind of bounce back up in this red dashed line where the model is on the left side, and then our actual temperature profile of that brine kind of doing that rebound after it starts to precipitate is on the right. And then this is just some other really cool pictures that we took. We don't really have a great system to measure this just yet, but we basically pulled out an ice core and then put fluorescent on the bottom, which is basically just a tracer that lights up in UV light. So the goal was definitely to look at these brine channels in the base of this ice core, which on the top right you can see super, super clearly as that fluorescent dye kind of permeates into that ice core. And then on the top left, you can kind of see where they actually emanate out of the bottom of the core of these two holes that are circled by the red circles. The bottom pictures are basically just like a visible light view on the left side, and then on the right side, you can hit it with this UV light. This was before we actually put any of the fluorescent on, and we think that the bottom is on the left side. It's actually just autofluorescence from the stuff that's in the in the ice. You can, definitely see a color gradient in this ice core. Some of the other things, the lake that I talked about that we couldn't really sample because it's probably like -3 or 4 C that day. So the problem with that is because these lakes are so salty, the ice, basically any of the salts and the brines that are trapped in it will start to just kind of dissolve and become, they'll start to flow very freely in that ice if it gets too warm. So when we went and tried to core the ice, you would get down a few centimeters and the brine would start to percolate up into your hole. So we kind of decided to make lemonade out of lemons in that case and do some like permeability tests that we hadn't necessarily planned for, which is where you basically drill to different depths within the ice cover, and then wait and see how quickly brine flows back into the hole to get a good estimate of how permeable the ice is. And this actually is a super useful number for these models, because in these models you kind of have to parameterize permeability. And it's kind of a difficult thing to measure super accurately just because it's describing kind of all the specific microstructure of the ice, which unless you have a fancy micro CT or something like that and can do some really detailed analysis on these systems, which is really, really hard to do in the field on natural ices, these are kind of difficult numbers to get at. So this was just kind of a way at looking or extracting these permeability curves to basically try and say, well, let's, get a first estimate of how well fluid can actually flow through these systems. So kind of the culmination of this is still trying to pull out all of these different profiles that we can get to kind of, get this huge catalog of these super diverse biological communities and chemical environments. Some of the group that we've gone up with, Alex Ponifract at Georgetown, is also doing 16S rRNA, so like microbial ecology on these samples. So we will, on top of knowing how much biology is in these systems of the different layers, but we'll know who is where in this system, and hopefully try and determine, are they active or are they just kind of in stasis and they are kind of just, prisoners in the ice and kind of at the whim of whenever it melts out. But we're really trying to get these huge comprehensive catalogs of all of these properties that we're interested in, things like the temperature, things like the chemistry, things like the water content, how much water is there, all this stuff that's going to determine kind of the habitability of the system, so that when we look at something like the results of our model, we can hone in on different regions and say, well, this is how, saline this environment is that we predict in one specific pocket or one specific region, so that we can kind of extend that to larger and more diverse planetary systems. And I think this is super important for understanding things like the geophysics of icy worlds as well as kind of like the astrobiology, just because the geophysics of these worlds is probably going to depend on what is in the ice as well, not just biology, but also salts and things like that. So understanding the diversity that can happen, this is an image of the surface of Europa. And so you can basically see like all of these different colors and features, and this is all just ice, but there almost has to be some kind of heterogeneity based on the colors and the features and things like that. So how do we go about understanding that? And it's probably going to be dependent on what's in the system. So knowing that from a geological or geophysical perspective is super important. But also, if you're looking at this image and then we're sending something like Europa Clipper, which is a big mission that's going to be launching soon for Europa, once you go there and maybe get even better measurements than this, you look at a picture like this and say, okay, well, where do we look for life? Where is it, this super colored region or this super broken up fresh region? It's really coming down to like, how do we determine, how do we predict these things? And it's going to come down to, where things are in the ice and how are they transported in the ice. And I think this picture from a chapter that Brittany wrote this last year is a really good example of how to go about that. And we're trying to basically use these models that can predict these evolutions of ice to simulate all of these potentially different systems on these environments and pick out exactly where we think will be the best place to look chemically, water availability, things like that, for different biomarkers and different habitable environments. So I'll just leave some pictures up here, and thank you very much. love to take any questions.